Hi there, welcome to the Basics lecture on gene regulation. While every cell has a full complement of DNA, only about 20% of genes in the genome are turned on in any one cell. Liver cells only turn on proteins needed for liver function, and eye cells only turn on proteins needed for eye function. How does this occur? The main mechanisms are these: chromatin modification, regulation of transcription, RNA processing, microRNA regulation, and protein degradation. First, let's look at chromatin modification. DNA is normally wound around histone proteins to form chromatin. A nucleosome is a unit of chromatin organization with 4 histone proteins plus the DNA wrapped around them. Access to DNA can be improved or blocked by molecules that bind to the histone tails or that bind to the DNA itself. Histone protein tails have charged R groups, so they tend to be attracted to each other. In histone acetylation, enzymes add acetyl groups to the tails of the histone proteins, shown here as little green balls. This makes the tails less charged, so they're less attracted to each other and the binding is looser. Thus, adding acetyl groups increases the transcription of that DNA region. In DNA methylation, enzymes add methyl groups, remember your functional groups from previous lectures, to the cytosine bases of DNA itself. Genes that are heavily methylated are more tightly wound and thus more inactive. Adding methyl groups therefore decreases the transcription of the DNA in that region. A large number of cytosines and chromatin are silenced by methylation, and this is one of the main mechanisms that prevents genes from being expressed. Once a region of DNA is accessible, the gene has to be turned on for transcription. This occurs at 3 different regions on the chromosome. Enhancers, or switches, are DNA binding sites that are often far upstream of the transcription site. Proximal control elements are DNA regions that are closer to the start site, and the promoter includes the Tata box and other elements, and it is right before the start site for mRNA. These DNA areas have binding sites for transcription factors. A transcription factor is a protein that has a binding site, shown here for DNA, plus an activation domain that can bind to other regulatory proteins or to DNA itself. Transcription factors can activate or repress gene expression. Activators is our term for transcription factors that enhance transcription. So regulation works like this. Activators bind to the enhancers and activate transcription. This triggers bending of DNA and the arrival of other, more general transcription factors, and all of these bind as a group to the promoter region. And this creates an active site for RNA polymerase to bind and begin RNA synthesis. So the presence of the correct activation transcription factors is required for RNA synthesis to begin. And it's this presence of these transcription factors that determines why different genes are activated in different cells. A liver cell has transcription factors that are needed in order to start transcription of albumin, but does not contain transcription factors for genes not needed in the liver. If you're in a lens cell in the eye, the transcription factors are available to turn on the gene for crystalline, but they don't have the activators that are needed to turn on liver proteins like albumin. Now let's move on to the RNA transcript. You've learned in the transcription lecture about how the pre-mRNA transcript is modified before translation. A cap and tail are added to the beginning and the end, and several segments called introns are spliced out of the transcript. The process of splicing introns is controlled by a type of RNA called small nuclear RNA. This RNA, called snRNA, is transcribed from the DNA. The snRNA combines with proteins to create a small nuclear ribonucleoprotein, which is abbreviated SNRNP or SNRP. Several SNRPs and other proteins then combine together to create a large RNA protein functional molecule or machine that makes an active spliceosome. And it's this that binds to the mRNA. The spliceosome has sites that match sites on the intron that's going to be removed, causes the mRNA to bend up. It removes the unneeded part of the mRNA molecule and then binds the exons together to replace it. Splicing is a way to regulate gene expression because different cells keep different exons during splicing. In this particular example, there are exons labeled 1 through 5 in the DNA. When the primary RNA transcript is spliced, some of the resulting mRNA has exon 3 but not 4, whereas other mRNA transcripts can have exon 4 but not 3. This produces different proteins because the mRNAs are different. Alternative splicing is controlled by activators and repressors that bind to the mRNA transcript itself and regulate which parts of the mRNA will be spliced out. Here's another recently discovered type of RNA involved in gene regulation. It's called microRNA or miRNA, and its function is to remove mRNA transcripts. Once miRNA is transcribed from DNA, it forms a temporary series of hairpin loops, binding to itself with hydrogen bonds between nitrogenous bases, just like DNA. The active microRNA region is tucked inside these loops. When the microRNA is needed, an enzyme called dicer clips off the loop of RNA that holds the strands together, and a protein binds to the microRNA, shown here, and removes the unneeded half that was next to the microRNA. Now, this protein plus microRNA complex can bind to the mRNA and either break it down so that it can't be translated, or capture it so that it can't be bound to a ribosome and prevent translation. In this way, even if a cell turns on transcription, translation does not need to occur. Lastly, completed proteins can be broken down after they're made. Many proteins are only allowed to remain active for a short period of time, hours even, and then they're tagged with ubiquitin molecules. And the ubiquitin acts as a signal that sends the protein to a molecular machine called a proteasome, which surrounds the protein and uses enzymes to break down the protein into its amino acids. We've now covered all 5 of the most clear and important aspects of gene regulation. It should be clear to you that while every cell has the full complement of DNA, the cellular environment strongly influences which genes are turned on and which proteins are active in the cell. This completes our basics video today. Thanks for watching.